get to know a little bit. Dr. Tim Sweeney, MD, PhD. I don't remember how long ago, but once upon a time, we were like in Mark Muson's class together, studying ontologies or something.
When I started the postdoc master's
we were
program.
talking about principal component analysis with in one of NIGM's TA sections or something like this. At the time, I think I was a fellow. You were a general surgery resident, is that right? But then in the middle of residency, you can expand into a research program. You found an attached profession, and then you were doing amazing work, all up and down. Go look at his publication of the record at the time. It was phenomenal. But then, I'm sure that's kind of what a lot of your story would be. It's like, wait, this is just a research project. There's a real thing that people could use and need, and it really needs a company to do that translation. And I think the world of academia could not contain the brilliance of what Dr. Sweeney could bring to the world. And he showed what was possible, and there is no overnight success, right? It's an overnight success after 10 years of hard work, I think is what it often manifests to be. Before we dive into that, could you just tell us about yourself? Where were you born, Tim? Where'd you grow up? How'd you end up on the path you're at, and how did you choose what to do in life?
Sure. I grew up in Colorado, did an undergrad at University of Chicago, studying neuroscience. Thought I was going to do a PhD for a while, ended up doing an MD PhD at Duke. Thought I wanted to be a neurosurgeon. Spent some time with neurosurgeons, found out they're all kind of insane, and so decided instead to go into general surgery. So I came to Stanford, as Jonathan was saying, for a general surgery residency, during which time I did the postdoc masters. I was here at the BMI program, did the postdoc masters 2013 to 2015, and so for those of you that I knew then, it's nice to see you all again, and we'll talk today about some of that path. What we've built since leaving that all started actually as my official postdoc master's sort of thesis project and grew into a company called Inflammatix.
And I don't know if you're naturally going to talk about it, but I don't know if this was always your vision, your intention when you were doing that training at the time, because I think a lot of people could learn from and be inspired by your story about how you navigated very big life decisions basically along the way.
It's not part of the talk. What I will say is, it was more or less the plan, and a funny story is when I was in residency, I went to a American College of Surgeons conference in DC. I was resident, and they were presenting at the time, this must have been in 2010, they were presenting on the GLUE grant, which for those of you that don't know, it's like a $100 million microarray project in trauma and surgery and burns. And they produced this paper that said that there was a genomic storm in an ICU setting, which meant that they couldn't make sense of what they were seeing. And I went to the microphone and I asked the guy, who was Ron Tompkins, who ran the project, and he was chair of surgery at Harvard. And I said, don't you think it would be useful if we use some of these data to do something like identify who has an infection and who doesn't? And he looked at me, I'll never forget this, and he said basically look, kid, the data are available. If you think you can do better, go ahead. And that was during my, it was actually before I was at Stanford. And so years later, we actually got that data and it became part of this project. And so the answer was, yeah, we did.
Oh, did you ever meet this person ever again?
No, although he did have to give us permission to use those data. So he was very kind in the end.
Okay, very good. Excellent. I think we got a pretty decent quorum now. We have over 80 people online as well. Please take it away, Dr. Tim Sweeney.
All right. Well, thank you very much, first of all. And again, it really is great to be welcomed back. I sort of put together a talk in 4 parts. And the first is probably what I'm most used to talking about, which is just what we built at Inflammatix and why it matters. I think it's useful to understand maybe the punchline, and then we can sort of talk about the origins of this project at Stanford BMI, and then also kind of what's next, how do we do science now moving forward as a company, and then I'll have a teeny piece on sort of starting your own company if it's something that maybe is in the back of your mind. When I was a resident in general surgery, our chair at the time was a guy named Tom Crummell, who was very tied into the biodesign program, and he used to say this: translational medicine comes in a box with a 1-800 number for sales and service. And that always stuck with me. It's one thing to sort of have a project that yields some neat papers, but the point is, unless you're actually helping patients at the end of the day, how much are we really accomplishing towards our goals? And with that in mind, I'll tell you the story about how we built a new tool to improve outcomes for acute infections and sepsis. And the typical place to start in sepsis is, what is sepsis? I think a lot of people have some notion of like, it's blood poisoning, right? And it's potentially deadly. They say to work on a big, hairy problem, sepsis is probably the biggest, hairiest problem there is in hospital care, which is to say it is the #1 cause of harm from misdiagnosis, it is the #1 cause of in-hospital death, and it is the #1 cost to Medicare, last measured at $53 billion per year, and that was pre-COVID. So big, outstanding problem. The problem is that in terms of it being a hairy problem, essentially every solution that has been thrown at sepsis has failed. There have been literally billions of dollars spent in either novel diagnostic solutions or novel therapeutic solutions. Many diagnostics have come to market supposedly for quote-unquote sepsis, and some of them are to measure bacteria in the bloodstream, except for lots of cases of sepsis don't have bacteria in the bloodstream. Others, for reasons I'll talk about, haven't been successful in changing clinical practice. And on the therapeutic side, there's been well over 100 drug trials trying to change the immune response in sepsis, and there have been 0 FDA clearances that were brought out of that. In fact, most people don't realize all of the TNF-alpha inhibitors, infliximab, rituximab, etc., all started when they found that TNF-alpha was high in sepsis, and they took those drugs to clinic in sepsis. Failed to make a difference in sepsis, but they said, hey, did you notice those patients with RA did better afterwards? And so went that story.
Like ibuprofen, somebody actually tested that?
Somebody actually tested it. It's
There are people
maybe that
testing
was the key.
statins at Stanford. I'm like, really? What are you trying? Anyway.
Here's the problem. My background was originally in clinical medicine, so I used to see these patients. And
Careful,
somebody
okay.
comes into the emergency department and they have a cough and a fever, right? They don't show up saying, this is sepsis. A cough and a fever can be a lot of different things, right? It could be a mild viral infection. It could be a bacterial pneumonia. It could be super bad infection that we call sepsis. It also could be a heart attack, or a PE, or an autoimmune flare. Like, there are a lot of things that look the same in acute inflammation, and so we need tools that aren't just unidimensional, right? If I say sepsis versus not, as a clinician, if someone said, hey Tim, the guy in bed 4 has not sepsis, okay, fine, but which of the other 17 things could this be? And so that's why I think prior tools for is it sepsis sort of never really took off. So when we started this project and founded the company, we wanted to build a product that would answer a lot of clinically actionable questions at once. And you can see them kind of down the left side there. Do I prescribe antibiotics? What other tests do I need? Does this patient have sepsis? That is actually really important right now because there's what we call a sepsis bundle of care that can only get to those folks. But most importantly, what's the disposition? Where are they going, right? The average ED stay is 3 hours. 3 hours. You have 3 hours to make the question about whether or not you come into the hospital, and everyone has read the stories about someone who accidentally got sent home and experienced a very, very bad outcome because they got worse the next day. So if we could build a test that would make a change in all of those things, it could potentially make a big impact in clinical outcomes. And the way we ended up doing that was by essentially creating tools to allow us to decode the inflammatory response. The fever looks the same. Somebody who has a viral infection or a bacterial infection or a post-surgical complication, a fever is a fever. A clinician can't tell the difference, but the immune system knows the difference. We've known this for years, that the immune system reacts very differently at a cellular level to a bacterial infection or to viral infection or to non-infectious inflammation. And it turns out we can also use it to risk stratify whether someone's going to get sicker tomorrow than they are today. So we built this test that looks at a panel of 29 mRNAs from whole blood. It's just bulk 29 mRNAs. And we apply machine learning to produce clinically actionable insights. And we built that into this test that we called Triverity. And I'll tell you a little bit about Triverity. The Triverity came through FDA with a breakthrough device designation in 2023 as a first and only test for an all-cause risk stratification for ICU level care. And was FDA cleared in January of last year and now is out on the market helping folks. And I'll play just a little bit of the video because I think sometimes it helps to kind of get a sense of the physical product. But one of the things that made Triverity really special is that it's very, very fast and very easy to use. So you take a tube of blood, this is a packaging tube, you just click it into this cartridge, you don't have to open the sample, you don't have to pipette anything, you don't have to centrifuge anything. It takes about 15 seconds of on-screen prompts, cartridge goes into the device and you walk away. So it's usable by a busy nurse and a busy ED. And in 30 minutes, you will get your results either on that little touch screen or in the thermal printer you see in the background, or of course pushed into the EMR. And what you see from the test is 3 separate scores. So Triverity is called Triverity because it produces 3 scores. There's a score that says, do you have a bacterial infection or not? There's a second score that says, do you have a viral infection or not? And those are separated, right? Because some people have a co-infection. You have the flu plus staph, and you can be high co-infected. Or critically, and much more importantly, lots of people come to the ED with a non-infectious cause of illness. And if we can say that's non-bacterial, non-viral, that's actually really helpful so that we can work through the differential. And then we have a third and separate all-cause risk stratification score that the official label is all-cause need for ICU level care within the next 7 days. And these 3 scores combine to do pretty interesting things. You can see this patient, for instance, high bacterial, high severity, that's a sepsis case. And in fact, the PPV on that is about 75%, much better than any other tool. But there's an anecdote I'll just tell you from one of the first customers that started using the test clinically. Within basically the first month of using the test, they had this guy come in his 60s who was short of breath, and he had a fever. And it sounded like a pneumonia. He had some sick contacts. And so when he came through the emergency department, they sent a typical, complete blood cell count, some basic metabolic panel, and a Triverity test. And the Triverity test came back as low bacterial, low viral, but high severity. And they thought, we thought this was a pneumonia, but now you're saying it's non-infectious. So they broadened the workup. They ordered an EKG and a troponin, and it turned out he was having a heart attack. And they said, thank God we weren't treating his heart attack with antibiotics. And he had a good outcome. But that's kind of the benefit of having these different scores and axes available. Now the test itself, the official label came from a 1,200 patient, 22 hospital study that we ran ourselves and was published in Nature Medicine in October of last year. It was actually one of 3 papers, and I'll tell you about the other 2 in a little bit. But sensitivity and specificity as high as sort of high 90s. The specificity on the ICU level care is 99% at the very high band. And as you've seen, it can drive a lot of clinical actions. When we make a product, we have to think about not just is it accurate, but does it make a difference in patient care or hospital outcomes, and ideally both. It's hard to build something that's actually onboarded and adopted, and so I'll just say that in addition to spotting infections early, which we have plenty of data, it's very, very easy to compare to things like procalcitonin or existing tools. We also want to be able to show that we can improve bed management in terms of admit and discharge decisions from the ED, and in terms of a diagnostic stewardship. If we could send fewer labs on these patients in the ED, they'll move through the ED faster. And I just wanted to show this because it's something we're particularly proud of. Last year we had 7 different sites report out real-world evidence of their experience with Triverity. And these are everything from 30, 40, 50 patient pilot series on top to several hundred patient IRB controlled studies to real-world evidence from their actual commercial use. And everybody's showing the same thing, which is they're able to send more patients home from the emergency department. Interestingly, what we actually show is that they send they admit a lot more patients if we say high severity, and they send more patients home at low severity, and that tends to net out to an overall increase in discharges. But 2 things for context. One, troponin, for those of you who are physicians, when it was first introduced, had about a 5 to 8% increase in discharges, so we're at least as good as troponin in terms of clinical impact in this setting. And 2, these numbers mostly come with a decrease in emergency department bounce backs. So it's not just that we're sending the people home, but they get sicker at home and come back. They actually more durably stay at home and do better. So that was what Triverity is, and I'll just say as a company, we are building other tests for other indications. We'll very, very briefly cover some of the endotypes, which is our therapy selection program. We have one in cardiac as well. But in the long run, the goal is really to sort of create what we might call precision medicine in a box, one that happens to have a 1-800 number for sales and service. With the idea being there's lots of diagnostic companies that you've all heard of that have driven really precision medicine, especially in oncology, right? These are the Foundation, Natera, Exact, Guardant, but all of them in a specialty lab format where you have to send your sample across the US and they do NGS and it's a 2-week turnaround time which is perfectly worthless in an emergency department setting. There's of course lots and lots of companies that build diagnostics for point of care use, but most of them are commoditized and relatively low value. And what we're trying to build in Inflammatix is what we might call specialty products, which is an ability to do these data-driven tests, but in an acute care setting, and really change the way we practice in hospitals. So I will move on now to talk about how it all started at BMI and what some of the data science is behind it. But before I do, happy to take any questions about Triverity or what we built in Inflammatix.
So off the record.
While being recorded.
While we're recording, sure.
Your Honor, example of the record.
Why is Stanford not using this?
We expect a pilot to get started here in a couple months.
Okay. Because I've been asking that internally for God knows how long. I was like,
top academic sites, and I certainly would never include Stanford in this next statement, but everybody feels that they are more special than the last. But I think that the weight of the evidence has gotten around to, it's time to take a serious
because I'm tired of implementing EHR-based sepsis predictors. I want to shut them down. And
it's amazing coming from you.
Every 6 months, a new one comes up, and I was like, guys, this is never going to work.
Did anyone see Joel Dudley, who came through the program a long time ago, once tweeted that sepsis prediction is the hello world of EHR research? And I think that's right. So anyway, yes, we hope to be moving forward soon. I think especially, just as a side note, we had built the product and it sounds really neat. I hope you think so, maybe you don't, but I thought it sounded really neat. And we had these hypothetical benefits we were pitching for a long time. Those 7 sites worth of data all reported out in Q3 of last year. And in Q4, we had 2 accounts move from first call to a closed contract in less than 3 months. So it's made a big difference now that we can actually say, your hospital will likely experience this as well. And I think that's been key. So, I thought this was neat. This really was, at the time, we actually stapled the first science translational medicine paper from the master's, to sort of the master's thesis paperwork and just said, here it is. Back in 2013.
Wow.
And you can see we both had a heck of a lot more hair back then.
He's so young.
Yes, it was very fun. Look, this is a great
No grace here, you see.
And I don't know how many of you are familiar with basically what Pravesh does over the years, but it starts with multi-cohort analysis, right? The idea that if you can pull together transcriptomic data sets from multiple sources, and in particular from all of the sources that we have available, we can pull out lasting insights because we're increasing the amount of heterogeneity we're seeing in those data sets. Now, you can sort of comment on this, but basically multi-cohort analysis had worked everywhere Pravesh had pointed it until I showed up in his lab as a young postdoc, fresh out of general surgery residency, and said, I'd like to study sepsis. And we kept trying to find durable signatures of immune response in sepsis, and it kept not working. And so I'll tell you a little bit about what that story was like and sort of the problem that we solved. First is to understand, and you sort of got a flavor of this, right? The idea was, we kind of know from basic cell biology that the innate immune system responds differently to different classes of insults. So for those of you that know the sort of the TLR receptors and those kinds of things, right? Your body reacts very differently and very early to a bacterial infection or to a viral infection or to non-infectious inflammation by kicking off slightly different patterns of inflammation, right? So, interferons for viral infection or NF-kappa B, JAK-STAT for bacterial infection, etc. And the basic idea was, look, if we have enough markers that sort of cover these different pathways, then we can use pattern recognition, we can use ML, to decode what it was that led to this particular pattern. So we can say, God, I see this pattern of immune activation, it must have been a bacterial infection. Simple hypothesis. The problem was that modeling acute infections is a very, very, very difficult task. Much more difficult, I would argue, than something like cancer, because there are 3 components of time that come into the data sets. And I sort of, they're here on screen, right? But there's the time since the infection, which is pretty straightforward. People show up sometimes 2 days after they feel sick, and sometimes 9 days after they feel sick, and that's a very different internal course of illness. There's time in the hospital, right? Sometimes we, do a study and we enroll, within 6 hours of ED admission. Sometimes we say within 24 hours of ED admission. Sometimes we say within 48 hours of ED admission. So that time in the hospital has changed that response, and of course sometimes there's iatrogenic infections too. And then there's time of sampling after treatment. So, sometimes, you get some notion of, well, we started with antibiotics in the ED, and this sample's been drawn late or early, or anyway, you can get a sense of how many different things are changing at once so rapidly in these patients. And typically what happens is one study is done with one very particular inclusion criteria, right? We are, as it says, we're studying community-acquired pneumonia 24 hours after admission. That was what we did for this study. Or, gosh, well, like the blue grant, we're gonna study trauma patients with hospital-acquired infections over time. Or, intra-abdominal infections right when they show up. So you get these kind of snippets, these little domains of a broader distribution of patients, and we're trying to learn across that. And I will say that the field itself had suffered from an enormous amount of overfitting. Typically what people did is they went to one of those data sets, we're gonna study community-acquired pneumonia, and within the data set we will learn a gene signature and we will cross-validate it and look at this, the AUC is fantastic. My goodness, we've solved this problem. And folks would say, great, can you test that in an independent data set? And they'd say, no, it's impossible because this is an adjuvant array and you want to test it in an Affymetrix array, and these technical differences are such that you just have to trust me, this is the answer. And so there were tons and tons of papers published that a gene signature existed for infection, and there was actually 0 attempts to actually translate this to a clinical tool. What we found was that the differences in acute care data sets dwarfed the differences between classes that we were trying to study. So these are actually real data. At the bottom, this is PCA or something, I can't remember. So on the left you can see data sets colored by study. These are individual patients. And so you can see the studies more or less cluster. And this, by the way, is across our 29 genes of interest. So these are just across dimensions that we know actually matter to the question of infection status. And in the middle, you can see them colored by class. I think blue is bacterial, yellow non-infected, and red is viral. And so, if you were, studying this and you got just the bright green data set, then obviously you're seeing a very, very small part of what you want in the end to learn. And this is sort of in this kind of hyperplane of separating red, yellow, and green. And in the end, then we also need a way to take what we learned from the transcriptomic space and reduce it to practice in a little 30-minute point of care device that doesn't use that same measurement tools. Now what we had done in the BMI program was essentially show that we could use multi-cohort analysis to select genes that were stably diagnostic across 3 separate domains. One was a question of what we call SIRS versus sepsis, which really is a question of is this infectious or not? And that was the first paper in 2015, Science Translational Medicine. Then we did 2 more, one for bacterial versus viral, and one to predict 30-day mortality in all-cause ED admissions. And if you actually just literally summed the gene sets that we published there, 11 plus 7 plus 11, that's a 29 mRNA set. And there was the idea that if somehow you measured these 29 mRNAs, you could build a product that would demonstrate both, is an infection present? If so, is it bacterial or viral? And could predict outcomes. And that was the idea we had that we fundraised around for Inflammatix. So at the time when we started the company, we just had this notion of these genes should somehow be integratable with a classifier and basically into a useful test. And we spun out. And over 8 years of development, we got that to market really across an iterative process where we said, we need to identify the right input targets. We need to develop a classifier and lock it, and then externally test it. But we also knew we didn't have enough data to really get to a point where this would be sort of clinical grade, so we iterated on that process. Lock a classifier, test it, take those data, push it back in. Lock a classifier, test it, take those data, push it back in. And over time, we got to something that we were happy with the performance, and then at some point, of course, we had to bridge the gap between, well, gosh, this works in RNA-seq, and we needed to work in the rapid platform. In 2020, so 4 years after founding the company, we published a paper in Nature Communications that showed for the first time a stable classifier that we could use across platforms in fully unseen patients to diagnose a single patient. So that's to say we, for the first time, had 29 mRNA vector as an input, and that for any new patient in a targeted platform, we could produce a probability that was actually accurate. And we, go ahead.
Sorry, its screen is gone. Are
Okay.
you still connected?
I don't know, the cable just came loose. Looks like it. I disconnected.
What about the wall? We have noticed it be finicky in the wall.
Oh, the wall,
might-
thank you, yeah, appreciate that.
Ah, the other end.
It'll come in.
Got it. Okay.
I won't go through this too much, but one of the things that we learned was how to treat fundamentally small data, right? If you look sort of at the top left, I know it's a bit of an eye chart, there's this kind of rainbow thing. Each one of those little slices of color is one data set that we had in our training cohort at the time. And the question was, how can we find something that generalizes well across different domains? And we tried a lot of different things, and sort of the flow-through graph is about both different ML algorithms, everything from sort of basic LR all the way through, as you can see, MLPs, and then also different ways to do cross-validation. Should we sort of randomly help people out? Should we leave one data set out? We could do it in different rounds. And in the end, what we found was that a very simple architecture of an MLP was a stable way to get these 29 mRNAs to produce probabilities that were much better than what existed in terms of existing tools, things like procalcitonin or CRP. And that we could actually diagnose one patient outside the data set and in fact move from NGS and microarrays over to a platform called NanoString, which is a kind of a high dimensional transcriptomic, but it goes from thousands to hundreds and is much cheaper to run. Over the subsequent years, we called that one, the algorithm BVN1, bacterial viral non-infection 1. And we had a severity algorithm too, and essentially this is a snapshot of a bunch of papers we published, and I bolded just the algorithm versions, right? So there was BVN1 and SEV1, and there was BVN2 and SEV2. And again, each time the idea was, we're gonna go to a new independent data set. We're going to find some patients, prospectively validate that this locked classifier works, take those data and then push them back into training. And from an incentives perspective, the way we did it is we basically said to researchers, hey, we've got a locked algorithm. We'll test it with you blinded in your data set. And that's a lot easier than going to somebody and saying, hey, why don't you gather some transcriptomes for me, I'm gonna do some discovery, right? Everybody gets a paper out of it, it's a fair test. And it also gives us a lot of confidence that individual sort of bumps in sensitivity and specificity aside, generally we were getting better, and also the algorithm continued to work. There weren't sort of groups of patients that just were falling out of what we were doing. And so that was sort of what was ongoing for about 6 or 7 years. But we also learned to transfer a locked signature with its weights between platforms, right? So if we had to do all of our discovery work on that little platform I showed you, the disposable cartridge, we would have had to get thousands and thousands of patients after development was completed. What we wanted to do was get thousands and thousands of patients before development was completed, so we could be sort of coterminous. And so we figured out how to take a locked signature that would be the exact same signature and use it in NGS, and use it in the platform, and use it in NanoString. These graphs are comparing individual patient probabilities across that rapid transcriptomic platform. We call it mRNA, play words on myRNA. So mRNA on the y-axis and NanoString on the x-axis, and our target was an R-squared above 0.9, and you can see we achieved it pretty much in all cases. So that was, then you had algorithms that worked durably, we had a rapid platform, and finally the sort of last step in terms of the ML was moving from, hey, this thing has really good, AUROCs, to all the other pieces you need in the end to get something clinical grade, which is, tight thresholds with, appropriate splines to create a unitless score from 0 to 50, and all the graphical outputs, and sort of everything that goes into taking from, this is a neat algorithm to this is really a production tool. And so for those of you who are interested, the final paper on the final algorithms is here. It was in ML4H. And the first author, Lumir Peturovic, was our VP of machine learning, who actually ran all of that process for all of that 8 years. And I think there's a lot of interesting tidbits in that paper if you go to take a look. What I'll say just generally is there are a lot of learnings for those of you who are interested in taking something, from sort of a paper through to a product. Number one, definitely, random cross-validation is entirely insufficient. Don't fool yourself. We need fully held out independent data sets, and I don't think that'll shock anyone in the room. We had to have domain expertise both in the clinical application and of course in the setup of the ML and lots of other areas too. How to build physical products and things. This is small data, right? Our final training data set was about 5,700 patients. It's not millions, not billions, but we were still able to make a lot of learnings and do something that others had not been able to do by using those data very carefully. And then I'll just say, yes, deep learning is cool, but especially in small data, it may not be necessary to build something that is really applicable. If this is of interest to you, and I'm happy to answer questions, finding variables, markers, mRNAs that both make good analytical targets and good machine learning targets took a lot of work, and we can sort of talk about that. FDA itself has a lot of new stuff now about good machine learning practices and how to build sort of appropriate controls around that ML process for IVDs. Lots of stuff we can talk about in terms of bias and fairness, et cetera, but maybe I'll sum up this kind of section by just saying the training we got here and what started here eventually is turned into something that you can hold in your hand and treats patients, and I think that was a really remarkable testament to this environment and the learning that goes into it. So let me stop there. I will sort of go to some future directions, but questions on any of the data science, machine learning behind what we built in Triverity.
And we got a few online as well, but let's let people in the room jump in.
If you were doing it again, what would you do different?
From a company perspective,
Or from your first paper here to where you are now?
The list is too long. We only have an hour.
The list is very long. I will say it was a 12-year journey from the first project to FDA, and that took a long time. We had to build two things coterminously. We had to build ML that we sort of believed in that would be clinically actionable, and we had to build a way to deliver it into the clinic. And actually, the way to deliver it in the clinic lagged quite a bit. I think that we would have had sort of a good enough, V1 that people could have used to treat patients a long time before we had a 30-minute turnaround test. And trying to find a way to lock a signature earlier, lock the targets earlier would have made that development a little bit faster, and maybe even potentially have a slightly less, less ambitious goal for that thing. It's 30-minute turnaround, no hands-on time, essentially. Maybe we would have been okay at 60 minutes. It's hard to know. We also, as a company, I will just say, I don't know if this is your question, but this was, if you go back and look at the very first pitch deck, what we built in Triverity is kind of right there, front and center, more or less as we had imagined it. But along the way, we did have a number of distractions that I think we could have, retrospect is always 20/20, but probably done without. I think it is remarkable that if you go back and look at that first pitch deck, you can see a drawing of a point-of-care device with a score, with sort of a stoplight color, and bacterial viral illness severity. Like a lot of that was pretty similar to what we had initially imagined, and I'm proud of that continued vision. Others?
You mentioned that you have these 3 time axes where the patient notices that something is wrong, has an infection, patient arrives at the hospital. Can you maybe go into a little bit more detail how you solved that problem, especially given that you only had 600 patients in your final data set? Was that enough for the
5,600 patients.
5,600. Was that enough for the machine learning to figure this out, or?
Yeah, it's a great question. So actually, I'll say first we tried to explicitly model it, and that just failed. There's just way too much going on. We dealt with it really in 2 ways. One is by removing some things that we just deemed clinically irrelevant. So for instance, There are a lot of data sets, if you look online, that would gather samples. You know, this patient came into the ED, but we allowed up to 7 days to take a transcriptome. And we just say, that's not helpful. It's only going to introduce noise. We'll just remove it. On the other hand, if we had a sample of within, you know, usually about 24 hours of someone getting an infection, even if they'd been in the hospital for a while, we included that. So I think the goal was, let's make sure that we understand the clinical circumstances we're going after and exclude things that don't match that and try to otherwise just include as much as we can. And at that, it's, you know, I want to say it's the unreasonable effectiveness of big data, but in this case, it was kind of the unreasonable effectiveness of small data. I think the other thing that we did, though, is that some of the ways we trained involved giving sort of special attention to different domains, to different data sets, to say, well, you know, if this is a strong outlier, let's pay attention to it rather than just pasting over that it's small and so it doesn't have a big effect in the final accuracy.
Maybe Ashish online and Tanya, they actually have similar questions about kind of risk, bias, how does it apply to different populations. But Ashish, do you want to go ahead and ask your question?
Sure. Thanks, Jonathan. I was great talk, Tim. I was just wondering how well would these transfer or translate over to a setting where the patients have different genetic makeup, like an Indian ICU setting, for example?
We have done prospective validation in obviously the lower 48, but also mainland China, South America, Eastern Europe, and Southeast Asia. So it does seem to be something that's pretty universal across sort of most of the genetic diversity we've seen. I think that's for two reasons. One is that in the end, we did have well over 100 different cohorts represented in the training data set, and I think they had some genetic diversity in them. But the other, realistically, is these are pathways that are deeply conserved, right? This is you know 100 million years of evolution for innate immunity, and so I think we just don't see a lot of variance in that across populations. One of the, I'll just add this, if you look online, we actually did, we wrote a paper on fairness and specifically looked at different aspects of demographics, and then actually had to recapitulate that for the FDA as well. Procalcitonin is the kind of standard biomarker that we compare against in bacterial infections. The procalcitonin actually has a marked underperformance in Black and African American folks. And what we showed was that the Triverity algorithms are essentially dead stable across different demographic subtypes, and I think it's because rather than sort of giving what nature imbues us with, we actually can learn something that's relatively stable across populations.
And a quick question on, has this led to any new therapies or drugs which were not traditionally used for sepsis becoming effective in sepsis treatment?
So I'll talk about that in this next step. So the short answer is I think it will, yes, but we'll get there in a minute.
We have more clarification questions online. Do you want to take them now, or do you want to go a little bit further? We'll cycle back. Gurmeet Saran, do you want to go ahead with the question? It's just a question about data sets. Go ahead.
Great. Hi, Tim. Thank you for the presentation. I'm not entirely sure if I'm understanding your data set collection correctly, but it looked like you were doing a retrospective analysis of these multiple data sets. And did those data sets, did you have blood sample, a biobank of blood samples available, or was the RNA signal already contained those 29 RNA sequences to get the information on? Were those already contained in the data set, or was it a hodgepodge and combination? Well,
For Gurmeet, nice to hear from you. It was, so we started at Stanford with all public data sets. That first, I think we had, across those three papers, maybe 1,000 patients from the public domain, Geo and Array Express and other places, just using existing transcriptomics. All of those prospective evaluations at the company, we always took the data and signed an agreement where we could use it for further training. So we grew a pretty large biobank over time. I said 5,600 were finally used in the training data set. The overall bank is well over, I think, 20,000 samples at this point. It's a little of both, right? We're happy to use something if it's in the public domain, certainly doesn't hurt, but obviously we wanted to build something proprietary as well, and it was something we could put some resources behind to grow it a little bit faster.
thanks, Dave. Appreciate it.
Great. There's a few other questions, but why don't you go ahead and we'll cycle back and just kind of let a thematic structure come back.
Okay, very good. So this is a tongue-in-cheek slide, right? But as an aside, when we get to the part on building companies, don't say you're a diagnostics company. You want to be like Tempus is, building a multimodal operating system for advancing molecular medicine, even if what you're doing is you're a diagnostics company. And we will talk about sort of number 3 here, therapy response prediction. We were fortunate enough to work with a lot of folks who are thought leaders in ICU medicine. There was this paper in Nature Medicine in 2022 that argued that really critical illness is due for an ontologic shift, where just like cancer used to be, you had cancer of the breast or of the pancreas, now you have a cancer defined by its molecular targets. The same is going to be true in the ICU, right? Right now, somebody comes in and we talk about somebody as having landed in the ICU because they were hit by a car, or because they have an infection, or an out-of-hospital cardiac arrest or general surgery. The reality is that may be true in the first day, but what keeps people in the ICU is critical inflammation. Right? What keeps them there is not an ongoing infection, it's the ongoing response to that infection. And maybe it's the case that there are different subtypes of that inflammation. And maybe that's why 100 drug trials for sepsis have failed, because just like you can't treat every patient with breast cancer with cisplatin, it turns out there are subtypes. Maybe there are subtypes of critical inflammation that would be separately druggable. And in fact, it might even be true that a patient with a car accident and a patient with sepsis have the same underlying subtype of critical inflammation, and maybe there is more similarity between these patients than we currently believe. So when you think about sort of a test for subtypes, what does that really mean? It's not a test for, it's not prognostic, right? The goal is not prognosis, it's prediction. What is somebody going to respond to? The goal is to identify distinct pathophysiology between these states, not just to separate them in some kind of meta state, but really something that we actually believe is linked to the underlying pathobiology of the disease. And of course, this starts unsupervised, right? Everyone, has a sense of what we mean here in terms of unsupervised in this room. But the point is, we can't say, here's a person who responded to this therapy and here's a person who doesn't, because you don't have the counterfactual. So you have to start simply in an unsupervised way to say, are there differences between these patients in, for instance, the transcriptome that are durable and true? There's no way to be wrong, right? If you publish that there is K equals 3 clusters in sepsis, there's no one can say, no, you're wrong about that. You simply have to say, well, there are these differences and it's reproducible, and maybe there are multiple different kinds of clusters. Well, this, of course, like cancer, has a precedent in how to sort of make sense of that morass. In 2015, there was this Nature Medicine paper looking at different subtypes of colorectal cancer. And at the time, there had been 6 different subtypes that had been sort of proposed and validated to some degree. And a paper came out and said, well, what if we tried to compare them all and see, is there a consensus or not? So we can stop saying use subtyping method A or use subtyping method C, but rather just they're all sort of telling us the same thing. And in fact, it was very successful, right? They came up and they said there are, in fact, 4 subtypes of colorectal cancer, and they are very different. And so as we move into therapy response as a company, one of the things we found is that ICU trialists are currently relatively loath to try any kind of particular subtyping in a clinical study, because there are literally like dozens now of different subtypes that have been proposed in the literature. We proposed one in 2016, based on work we've done here. But we were one of about 6 groups that had done so using transcriptomics. And so the idea was, maybe we could fund a consortium at the company and compare existing subtypes and see, in fact, if there is a consensus. And so I thought this is an interesting story just because it's something that you can do as a company that probably we wouldn't have been able to get funding for. So we founded this consortium, subtyping in sepsis and critical illness, called Subspace. We signed 11 academic partners, and the first 5 or 6, we basically said, look, if you give us your samples, we will sequence them and give you the RNA-seq data back for free. You keep a copy, we keep a copy, and we'll bring it into this kind of main data set, and we'll all share in the main data set. And the number one thing is we're not going to use this pooled data set for discovery, just for validation. If you have an existing signature for subtyping critical illness, we'll apply it, We won't do any discovery, so there's no new IP you have to worry about. And we're only going to use retrospective samples because lots exist. And it was pretty successful. In the end, we got 11 academic partners from pretty good places, over 10,000 samples. And we said what we're going to do is we're going to compare subtypes of critical illness and see if there is a consensus. And in the end, it was Inflammatix funded, but Pravesh led a lot of the analyses. And I'll just say that was the other two of the 3 Nature Medicine papers we published last quarter. It does turn out that there is, in fact, a consensus among these different subtyping methods in terms of what immune dysregulation looks like. And I didn't have time to go over the science in the talk, but a pitch for next week is I think Pravesh is on track to talk about that science and sort of walk you through it. What I will say is that as a company, we can think about how to build data sets and how to build value with sort of 3 legs of a stool, right? And the number one is we have to start with something of actual clinical value to a customer. And right now that's Triverity, right? The sort of the lower left here, the diagnostic piece. We've got a transcriptomic point of care device delivering value in the ED, and it happens to use PAX-gene tubes, which are RNA stabilized tubes. And it turns out that remnant samples are obtainable in sort of easier ways than prospective studies. And so we can begin to collect data at commercial scale in terms of the transcriptomic outcomes. Step 2 is the slow but sure integration into the EHR, where both the customer sees a benefit in terms of, oh gosh, Triverity's there and I can use it for things like quality and billing, etc. But we can also then begin to pull linked clinical outcomes out of the EHR and build what we think is probably the world's only emergency department to ICU clinical transcriptomic data set. And that kind of leads to part 3, which is, as we've seen in other areas of precision medicine, ultimately the onboarding of novel either indications for existing therapies or novel therapies for ICU medicine, because we can find and generate these subtyping signatures and actually deliver them into the point of care. So that's kind of the vision of what we want to build in the long run in Inflammatix, and I'll just spend maybe 5 minutes talking about building a company at the end for those of you that are interested. This is not my quote, but I think it's pretty accurate. Founding a startup is like eating glass on stage for a prize that may or may not come. You can quit anytime you want to. All you have is a belly full of glass. Moving forward with an idea, whether you're doing it in academics or in industry, you used to rejection. I went back and looked. So in 2013 and 2015, we applied for 9 grants. Most of them were pretty small. Of those 9 grants, we had 7 failures and 2 successes, totaling $55,000. I pulled out a couple of the quotes. Neither the study design or the approach used is innovative. I like that one. And the host response approach for sepsis assessment is not sufficiently convincing, which, it probably wasn't at the time. We didn't have a lot of data. When I was a postdoc, we actually briefly tried to license the idea. This looks like it should be able to help people. But, I'm this postdoc, and I was going to be a doctor, and I don't know about how to build businesses. So we'll go to Roche, and we'll go to, Abbott, and we'll have them build it. Mm-hmm. And the reality is we went to them, we said, hey, if you pay us money for this thing, and you hire a team of people you don't have, and build a device you don't have, then you can disrupt the markets that are sending you billions. And shockingly enough, the answer was yeah, it's not really for us. So we decided to do a startup. And we decided to do a startup mainly because I had applied for a grant, actually a Burroughs Wellcome Transition to Independence faculty grant that got rejected. And it basically said, this doesn't look like science to us, it looks like product development. And anyways, if it was such a good idea, someone probably would have done it already. And it took a long time to calm down and then said, at least with an investor, you can go talk to them. Right? It's hard to submit a grant and address every single question. And so we did. And actually the business first got funded with a DARPA SBIR, and then we got our first round of funding committed from Coastal Ventures, and off we went. 2017 to 2025, Inflammatix applied for 26 grants and contracts and won 9. So the acceptance rate went from 22% to 33%. It's not a ton. What I will say is we pulled in over $40 million in grants and contracts. So the amount of resources you can get in a company is very different. And what I will say, number 2, is that if you don't like grant funding rates, just wait until you start talking to investors. They are much, much worse. Now, Inflammatix, you know, look, we were at the right time at the right place. Stanford is an amazing place to come out of. Data had been accumulating in terms of, you know, all the MAQC and the NIH protocols that you had to publish those transcriptomics. Atul, bless his soul, had come up with how to knit together some of that transcriptomics, and Pravesh worked with the tool, and then Pravesh had come up with the multi-cohort analysis. We had gotten pretty lucky with the initial Science Translational Medicine papers, but the broader market was there too. The ability to actually say, hey, we are going to be able to measure 29 mRNAs in 30 minutes didn't seem outlandish. It wasn't done, but it wasn't impossible based on where the technology was. There were market pull incentives. Sepsis was beginning to be something that CMS was focused on from a value-based purchasing. And then this is true for all of you as well. Capital was available, right? Diagnostics had recovered after all the nonsense with Theranos, and we were at Stanford, and so were you. Sand Hill Road is very, very close, and for those of you that aren't aware, Coastal Ventures literally sits catty corner from the tip of the Stanford golf course. I mean, you are closer to venture capital than anybody else in the world. Now, spinning out a company has a lot of things, and actually funding is somewhere sort of in the middle. Number one step is deciding to do it, and then you kind of have to go through, what am I doing? And I have to learn to pitch it, and you know, honestly, your whole job for the rest of your life in CEO world will be sales, but that's okay. If it's a story that you're passionate about, you won't mind it. You have to find a team, and you have to license your IP out, and you have to, you know, get some grants and talk to investors and go through an accelerator and everything else. All of this is accomplishable. There is a playbook. It's an entirely learnable process. It's really fun, it's really hard, but it's things that you can do. And in the end, you know, it's all about people. Right? It's about finding people to work on your vision. I am constantly amazed that we said, hey, we should build this product, and people dropped what they were doing and came and joined it. And you know, at some point in 2022, we had a working clean room where people were building cartridges, wearing like bunny suits and stuff. Like, it's pretty cool, right? Went from, you know, some code in a computer to like a clean room building products. It's all about people, and we've been enormously lucky to work with those people. I will just say, don't forget about family if you do this. My first son was born 2 months after Inflammatix was funded. He's 8 now. It's been a nice journey. And all of this is leading to this final soapbox slide, which is this. And I won't make you raise your hands and out yourself as someone who might not be interested in academics. But for those of you that are thinking that it would be pretty fun to take your project and found a company, do it. You are literally at the most renowned institution for spinning out startups. You work with the absolute best professors who've taught you the absolute best things. You have the best, most collaborative peers. You know, you are super close to funders. If not you, then whom? So I'll just, you know, it's really, really fun. You will have a great time. And if anybody does want to talk about it, I'd love to always chat with folks who are interested in founding the business, or maybe even further along. Feel free to ping me there. And I'm happy to take your questions. Thanks for your time.
All right. Any comments or questions in the room?
Great talk. Thank you so much. You mentioned you are collecting many data sets and you wanted to train based on this collection of data sets. My question is that when you are using different data sets from different population and instances, have you ever tested, for example, the uncertainty of the model when you train based on different samples? So how models certain about prediction when you are using different data sets, and specifically when the sample size is limited and the feature size, I don't know what's the multi-dimensionality, the number of features. In the conditions when dimensionality is high and we have a limited, for example, data sets, this uncertainty sometimes has a great impact on the prediction. Have you ever tested this?
So to answer a couple questions. First, we have a 29 dimension. We measure 29 mRNAs at once. And as a side note, the reason we didn't end up changing that was twofold. One is we actually did some early experiments and said, well, what if we went up to 500? And it didn't really improve performance. And 29 mRNA became kind of like a generic for the product that we could publish around. We did a lot of work on uncertainty for a while. I will confess I have not done what you have asked, which is what about if you look at uncertainty in particular in new domains and does it go up? That's an interesting question, I don't know. We thought a lot about whether it would help a clinician to show an uncertainty score, and in the end, after talking to a lot of clinicians and doing some market research and thinking about the regulatory implications, decided that even if we knew it was uncertain, it wouldn't change what we actually presented to the user. So, we sort of give a score from 0 to 50, and rather than say, well, it's 0 to 50, but we're more or less certain here, we just sort of say, this is the score. And I think that's because, physicians are already overwhelmed with a lot of numbers. They already don't quite know how to use a new test. And if you're saying, well, it's 49, except for it might be something else, it sort of makes it even less trustworthy. What I will say that's interesting is that the test is often very certain where clinicians are uncertain, which is generally good, this one on. But it's also interestingly sometimes very uncertain in cases where actually the physician was already certain. It's pretty orthogonal, and so I think it's something that we'll need to continue to study over time.
I have a couple, and Brian Bunny, if you want to come in next. Can you describe just procalcitonin a little more, just as a reference point, and why people liked it or just didn't do the job, and why this is different? And it's also coming back to, is this a data science project, or was it a molecular project, or a product project? Like, really, all these things really had to change, and how did one work and not the other not?
So for everyone, procalcitonin is kind of the existing biomarker for detecting bacterial infections. It was initially used, was thought to be a good marker of cancer, and then they looked and said, oh, look at this, it's actually mostly up in patients who have bacterial infections. And so it was brought really to market in the early 2000s by a company called Brahms that was then actually being bought for a decent price by Thermo Fisher. And now it's sort of in use, which is to say, especially in Europe, it's used a lot more. Here in the States, it's very rarely used in the emergency department, a little bit more in the ICU. The number one problem with procalcitonin is that it's from what I'll call sort of the prior era, which is the era of discovering biomarkers, right? Somebody found procalcitonin and said, gosh, look at that. It's correlated with bacterial infections. Let's go make a product out of it. And nature had imbued it with some powers, and it was going to be as sensitive and specific as procalcitonin is. It's actually a propeptide, so some people release procalcitonin and some people don't. And the number one problem with procalcitonin is that the sensitivity for bacterial infection is in kind of the 70s, maybe 80s. So when you have a negative PCT, procalcitonin, you don't necessarily know that that patient definitively does not have a bacterial infection. It's decently specific. We are in a new era, right? We are now in the era of engineered biomarkers, where we have to pick what we want to do, right? We're going to pick, I'm going to say bacterial infection, and I'm going to see how many markers are in it. I'm going to try to engineer some particular sensitivity and specificity. And we actually targeted very specifically a 97% or greater sensitivity in the bacterial score and hit that in the final piece, because we basically went to the market and said, what do you want? And I think this is going to be more and more true, right? That yeah, it's cheap to produce a single biomarker, but we can do better than sort of nature does, right? In algorithmically composing different biomarkers to create a set. And so I think that's been one of the major advantages, is basically saying, what does the market need? Let's go design it, rather than, hey, there's a bright spot on this gel. That's the tool we're going to use.
And compose. I have a lot more rich stuff we can talk on, but I'm going to let a student get the last common question. Brian Bunning, if you would like to unmute and go ahead.
Hi, yep, I'm a fifth-year PhD. You said at the beginning that in Q3 you had hospitals give you data, and so I wanted to ask, how has adoption been? Like, does this technology sell itself, or have you found a new struggle in the sales, even though you have this vetted technology that seems pretty clear that NIGM wants to have in the hospital?
I will answer a little bit tongue-in-cheek. A startup is basically saying to yourself for years, after that milestone, things will get easier. and then you pass that milestone and they get harder. Hospital sales it's multifaceted, it's not easy. They're very large institutions that move slowly. What I will say is, with clinical utility in hand, it is picking up very rapidly. And we actually hit more or less our sales target for last year, which was an ambitious target, and we appear to be on track for this year, given the 3 weeks that we're in. I think more to the point is, in the end, it's not enough to have something that seems like it will help one person. We have a benefit for the clinicians that want to use it, right? It's going to make your clinical practice better. It's going to sort of save you from making a wrong decision. We have a benefit for hospital executives in terms of bed management and the cost savings. And we have a benefit for the lab in terms of it's easy to use, and we might be able to decrease the number of diagnostics you run. But any one of those patients, or excuse me, any one of those physicians or groups can block a sale. And so it's necessary really to make sure that you're trying to align incentives, not just for, hey, this looks neat to a clinician, but also everybody else that's part of that sale to bring it in.
Fantastic. I'm sure a lot more we can learn, and maybe people can approach you directly for those in the room, or for those online. Thank you so much, and exciting work and showing that journey, and we'll hear more from Profesh next week. Thanks, everybody, and thanks again, Dr. Sweeney.
Thank you.